All right, well, welcome everyone. Welcome to our newest webinar where we're going to be introducing Neurolucida 360's enhanced batch pipeline. My name is Shane Baldwin. I'm a software specialist stationed out here in Salt Lake City, Utah, though all my colleagues are back in Williston, Vermont at the MBF headquarters. They just got a really big snowstorm that I'm jealous about. So I'm sure we'll have a couple more people join in the next couple seconds, but just kind of what we're going to do today is run through what's included in our new enhanced batch pipeline, how to use it, and then at the end we'll kind of do a little Q&A with product manager Macy Heel. So along with our batch pipeline, we're excited to announce that the newest release of Neurolucida 360 was released on Friday, December 9th. So I hope some of you already have some of that experience in the new release. We're super excited about it and super proud of it. So let's get started. So at any point, if you have any questions during the webinar, there's a little Q&A button at the bottom of your screen. You can go ahead and click that, type your question, and send it on over. I'll try to answer any questions I can during the webinar, but if I don't, I promise we'll get to it after in our little Q&A session we have with Macy. And that Q&A session isn't necessarily just limited to the batch pipeline, but also any other questions you might have about the newest release or general questions about Neurolucida 360, because I know not everyone here today will have the experience of using it before. So those who are getting kind of their first look at Neurolucida 360, welcome. So what's included in our new batch pipeline? So you have the ability to use image filtering and tracing in one workflow across multiple images. And what's included in the image filtering? So we have our Golgi filter, our projection filter, our in vivo filter, our vessel filter, and our closing filter. And we'll go into a little bit more about what those are in a second. And then we have our automatic tracings settings and methods as well. So we have our soma tracing, our tree tracing, our spine tracings and spine classification, our puncta tracing, and varicosity tracing. So all of this is included in the batch pipeline. If you have any more questions about general tracing, please check out our webinar titled Introduction to Neurolucida 360. And if you have any more questions about maybe seeing some of our image filters in use, we have a webinar called Applying Image Filters to Improve Reconstruction Accuracy. And that would be a really helpful resource for those who have more questions on that. Both of those webinars can be found on our YouTube page. So just go, once you get to YouTube, type in MBF Bioscience, you should be able to find us and find those webinars. So quick review of our image filters in our batch pipeline. So we have our Golgi filter, which is going to cut down the out of focus shadows that are the result of labeled structures in the tissue. For those who have experience with Golgi stains, sometimes there is some Z blur in the tissue, and this Golgi filter kind of cuts that out. We have our closing filter, which is going to perform a grayscale morphological closing of the image. And this is useful for closing small gaps inside of a labeled structure. We have our vessel filter, which is going to identify boundaries of vessels and other hollow structures and make them appear solid. How that helps is that when you have a hollow structure, sometimes our tracing algorithms aren't as good, but this closing of those vessels is going to help you be able to trace those in Neurolucida 360 if you'd like. Our in vivo filter is going to be designed to remove motion artifacts caused by consecutive in vivo acquired images. And then our projections filter, which is just maximum and minimum intensity projections, which I'm sure a lot of you have experience with either in our software already or you just understand the concept. That's a pretty pretty common one. So one more overview of what reconstruction and automatic reconstruction methods we can do in Neurolucida 360. So we have our soma reconstructions, our trees, our puncta, and our puncta can be reconstructed by themselves or in proximity to other structures that you trace. We have a varicosity reconstructions, and lastly our dendritic spines. So we'll get a good look about how the batch pipeline can work with a lot of these at once. And on that note, for today's webinar, we have a little example data set. So say we have 3 groups that we want to reconstruct and analyze, but these 3 groups of images that we have are from different types of specimens with different types of imaging and or staining. So our group 1's we want to filter, reconstruct, and analyze Golgi stained dendritic specimen samples. Group 2, we want to reconstruct and analyze soma, dendrites, and spines, and that's in confocal imaging. And then group 3, we want to reconstruct and analyze dendrites and puncta in 2 different channels. So we want to do all of this at once, and we're going to utilize the batch pipeline to do so. Before we get into using the batch pipeline, we do need to save some 3D settings that we can take advantage of in the batch pipeline. To do that, we're going to take one representative sample image from each of our 3 groups, and then save the corresponding optimized reconstruction parameters, and then use them in that batch pipeline. So just a quick example of this, we'll reconstruct one of our neurons in group 2. So very quickly, we're in a 3D window in Neurolucida 360, and we'll first start by reconstructing our soma. And then we'll use our automatic tree detection parameters to detect the dendrites and axons in this image. So here we're going through and optimizing those tracing settings. We'll increase our sensitivity here for this image. Go through and validate some of our seeds that are being placed. And then lastly, kind of clean up and optimize some last tracing settings and parameters. Then we'll go ahead and trace our trees. And lastly, once we have our soma and trees traced, we want to detect and reconstruct our spines on this image. So once again, optimizing our spine detection parameters, go ahead and detect those. And then we'll use our automatic classification feature, classify all, and then we have classified and detect spines on our specimen. We'll go up to this icon here. This is our 3D settings icon, and we can save these settings that we just made. And we'll call them group 2, because this is what we want to be using with our group 2 images in the batch pipeline. So I hope that was clear and how we would just save our settings to use in the batch pipeline. If you have any more questions about that, please feel free to ask in the end. But now we want to move on to using our batch pipeline to go and trace and or filter all of these images in each of our groups at once. So first thing I want to point out is our new pipelines ribbon in the 2D window. So this is a brand new little space we created for our batch pipeline. We also have our detect cells workflow there and some other workflows previously in Neurolucida 360, but I wanted to point out that this is our new pipelines ribbon. And first icon on the left there, you can see that our batch pipeline icon is right there. So first thing we want to do is add all of our images. So we'll go up to add images. we'll go to our batch pipeline folder that we have with our 3 groups, and we'll go group by group and add our images. So group 1, cells 1, 2, and 3. You can see that there's a little G at the end of each image name here, and that's just reminding us that we want to run a Golgi filter on this group. Next, we'll be loading in our group 2 and 3 images. And once we do that, we can go ahead and get started in setting up our batch pipeline. So all of our images are in the window now. You can see that each image has a status of setup required. So we're going to go ahead and set our batch settings. You can see here our image filter drop-down menu. Nothing's there now because we haven't set anything up, but we'll go ahead and go to setup. We'll create a configuration with the add configuration button. And just because we're only going to do one image filter, we'll call this group 1 Golgi to set up our Golgi filter for those 3 group 1 images. This is where you can choose filter type. Ours, we're going to choose Golgi, but you can see the rest of our filter options are in the same little dropdown. It's going to mention that the resulting filtered image is going to have a output label suffix of _Golgi. You can set whether or not you want the resulting image to be compressed. And then you have some Golgi settings. We're just going to use the default today. And you always want to make sure you have your bright field image box checked when you're working with bright field images. We can go ahead and save those changes and select return the list, because this is the only image filter that we're going to use in today's example. Next, we want to set up 3 different detection settings configurations. Why? It's because we have 3 different groups where we want to trace 3 different types of detections. So we're going to set up group 1. So we'll add configuration and do group 1 trace. Our group 1 saved 3D settings from before are already there. And if you remember, for group 1, we want to trace trees and spines in our Golgi image and also run spine classification. Go ahead and save those changes. And our group 1 detection configuration is all set. So we're going to do this 2 more times, specific for group 2 and 3. For group 2, same thing, we'll name it so we know which one we're picking. We'll select our group 2 settings that we set up beforehand. And it's the same things that we want to trace as group 1, except we want to add our soma detection as well. Let's go ahead and save this. Last but not least, we'll add our group 3 configuration. We'll call it group 3 trace. We'll make sure group 3 is selected in the dropdown. And then for group 3, we just want to select tree tracing and puncta tracing. So we'll uncheck spine classification, and soma, and we'll check our puncta box. We'll go ahead and save these changes. And now we can return the list and start applying these settings to our images. And how to do that? Well, first you have to highlight the image you want to work with. So we'll highlight our cell 1, and we know we want to put a Golgi filter that we set up and our group 1 detection settings. So we'll look at the drop downs and we'll select group 1 Golgi along with group 1 trace. And now you can see that the status went from setup required to ready to start. And now we want to do this for the rest of our images, but we can do this in bulk. So you can go ahead and select the rest of group 1, cells 2 and 3, and select the same image filter and the same detection settings that we set up. Once we've done that, we can go ahead and hit apply. And now we can see that group 1, all of group 1 is ready to start. So we'll do the same thing for groups 2 and 3. We'll select multiple, We'll select all of group 2. We don't want to add an image filter for this group, so we'll leave that blank. And then we'll go to our group 2 trace detection settings and select those for our group 2 images. For group 2, we know we're only working with one channel, so we can keep the default of detecting these on all channels. In group 3, that's a different scenario. So you can see group 2 is ready to start once we've hit apply. And now for group 3, we'll do something similar. We'll ignore our image filters for now because we don't want them in this group, but we'll go to our group 3 trace. And in group 3, we want to run this on the detection settings on multiple channels. So we know it's a 2-channel image, so we type that in. We'll hit refresh display. Now we have an option for channel 2 specific tracing. We know our puncta are in channel 1, and our trees that we want to trace are going to be in channel 2. So we can specify that in our little window here. Go ahead and hit apply. And now all of our images are ready to start. So go ahead and click start. From here, you will get a status update for each of the images, whether it's filtering or tracing. You can see that the current status for cell 1 is running Golgi on all channels. So it'll do that first, and then it'll detect on that resulting Golgi filtered image. But for the sake of time, we'll continue on to the batch analysis portion. So the resulting data files that you're going to get out of your batch pipeline, you can take those XML files and load them into Neurolucida Explorer's batch analysis feature. For those of you that don't know Neurolucida Explorer, it is the analysis software that accompanies Neurolucida 360. And here we have our cell 2, or not cell 2, our group 2 cell that we traced earlier. We want to go ahead and not only analyze this image and this tracing, but the rest of our resulting XML files from our batch pipeline. So we'll go up into the Analyze tab and select Batch Analysis. It's going to ask us if we want to include other files, which of course we do. And we'll go back to our batch pipeline folder that we had created earlier. And instead of going folder by folder, and we know all the resulting data files are XML, we'll just search for our XML files here. We can go ahead and select all. And this is going to now load all of our resulting data files from our batch pipeline in Neurolucida 360 into our batch analysis feature in Neurolucida Explorer. So there are multiple files from multiple folders, so it's going to take a second, but it should pop up here in any moment now. Here we go. So now we have our batch analysis window. On the left-hand side is going to be all available analyses that we can run. If you hover over each of the analyses, it'll give you a little analysis description in the top right corner. That's helpful if you have any questions on what you're running, or if you forget, which should I use, individual tree or each tree, and then it'll tell you kind of the general idea of each one. So we want to add a puncta analysis. We added a tree totals analysis. We'll run a shoal analysis and a spine analysis for all of our images here. We can choose save options, so we can separate by set. We can choose one Excel file for each data file, or we can choose one Excel file for each analysis. That's on all personal preference. Then you can choose where to save these Excel files when this is complete. So we're going to choose our batch pipeline folder once again, so everything's in the same place. And then you can choose whether to open these Excel spreadsheets upon completion. So we'll check that, and we'll hit run. So when you do hit run and you do have open Excel spreadsheet on completion checked, it's going to start opening all of these Excel sheets at once. So we don't have to currently deal with all of that in the window. I'll just open up a couple of the resulting Excel sheets. So if you've exported anything in Neurolucida Explorer before using Excel, this will look really familiar. But even for individual files in Neurolucida Explorer, you can export whatever analyses you use into Excel for any further analysis or figure making that you would need. All right, so this concludes the end of our batch pipeline. And just to recap, so we had 3 different groups that on some images we ran filter, and then on all images we ran automatic detection settings to trace different features depending on the group. From there, we loaded all the resulting XML files into Neurolucida Explorer and took advantage of the batch analysis feature in Neurolucida Explorer to run analysis on all 9 XML files that resulted from our batch pipeline. So just a quick shout out, we'd like to thank the investigators who allowed us to use their microscopy data in today's webinar. And with that, we'll take any questions that come up. And I'll welcome in NL360 or Neurolucida 360 product manager, Macy Heel.
Hi, everyone. Hi, Shane. Great job. Like you said, we're really excited that Neurolucida 360, this latest version, is out in the world and ready for all of you to use for your projects. I hope that this new and improved batch pipeline will help you streamline your data collection and help you get the answers you need faster than ever before. So let's take a look at the questions that are coming in, Shane, and we'll hopefully get to all of them. All right. So a question here: does each file need to contain only one cell, or can it contain many cells? Each file can contain many cells. Or just one cell. The detection algorithms will find many somas and trees from all present cells within the microscopy image that you've imported into Neurolucida 360. There are ways you can, and this actually kind of ties into another question we have of, can you edit the reconstruction after it's run through the batch pipeline? And the answer is yes. So you can go in, take one of the XML files that Shane, you showed in Neurolucida Explorer, bring that into Neurolucida 360. Before you go to analyze, just make sure the tracing is accurate. You can edit it point by point. You can use the semi-automatic tracing functions to clean up anything that was missed. You can also, at that point, discretize between the different cells. So you can select objects, so a soma and the dendrites and the spines from one cell, give it a name, say this is my cell 1, or maybe this cell relates to this ePhys data, however you want to kind of classify those cells, and then once you've made those adjustments, you can then use the batch analysis pipeline that you described, Shane. All right.
We had another question from John. I use both NL360 and VL360. Will this batch functionality be in VL360's next release too?
We'll clarify first, VL360 stands for Veselucida 360, which for those of you who are here and don't know about, it's our vessel tracing software and analysis software. And the answer to John's question is yes. The next release of Veselucida 360, which is coming anytime here early in the new year, will also have this batch pipeline in it. It'll be brand new to that software, so you can make use of that great vessel filter, enclosing filter, and the automatic algorithms that are in Veselucida 360 to do this type of streamlined analysis. Let's see. I have another question here. How do I know what settings to use for the Golgi filter? So it looked like, Shane, when you were giving the demonstration, you set a sensitivity setting and an edge clearance setting for that filter. And we've designed the batch pipeline to allow you to not only conduct a multi-file processing workflow, but you can also leverage the tool to find the best parameters for your data. And how you do that is you would open one image that you want to test those settings on for your Golgi filter, and copy that image within the queue of the batch pipeline like you had for your different images, Shane. And in this case, you can run different sensitivities or edge clearances across the different files in your queue, let the batch pipeline run, and then you can easily open those images in a grid view in Neurolucida 360 and pick which sensitivity works best for your image. Where you've removed the most background and have your spines and your dendrites really clear and visible for the reconstruction algorithms. Oh, what else do we have here? What is being applied first, the filter or the tracing? Do you want to take that one over?
Yeah, I can take that. So yes, in our case, in group 1, we had to apply that Golgi filter along with the tracing that we wanted to have. And what happens first, and if this, you can see this, I apologize for not showing it, but the XML file that's written will actually have that underscore Golgi suffix, yes, suffix to the end of the file name. And what's that telling you is that it's running the filter first and creating that new image, and then it's running the detection on the newly filtered image. That way, you're not creating, if you wanted to do both, you don't have to load in the images twice. So that resulting XML file, that resulting tracing is going to be done on the filtered image.
Precisely. And then lastly, can I try Neurolucida 360 out on my data? And the answer is yes, of course. We would love to talk to you about your data, provide you with a free trial so you can evaluate if it will help you in your research. Our team, Shane and I specifically, are available for giving demos of the software as well, and doing evaluations here to provide you with tips and tricks for getting the best reconstruction with your specific image data. I think that, unless anyone else has any questions here, I think we've answered all of them, and
we are about 1 minute from when we said, oh, we had one more question come in.
After batch tracing many images, can you edit the results in Neurolucida 360? Yes, you definitely can. And like I mentioned before, you can do lots of different types of editing with the very robust editing tools in Neurolucida 360. You can disconnect or connect branches. This is just for the tree editing. You can adjust the size or the position of the points. You can merge or split spines. You can do a lot of different types of editing to make sure you're getting down to ground truth.
All right, well, thank you everyone for taking the time today. We're hoping to have a couple more of these out soon. I know we haven't had a webinar in a while, but we really appreciate everyone taking the time to join us today for our introduction to our batch pipeline. So if you do have any more questions, you can please contact us at info@mbfbioscience.com.
I hope everyone has a happy new year.
Yeah.
All right.
Happy holidays, everyone.